Thanks so much, Brad. It's not true, as you'll see soon. um, with such a nice setup like that, I don't know what to say. I'm really pleased to be here. I think, is this a little too loud? Maybe, okay. Is it good? So I'm really pleased to be here, and I really enjoy EHIP every year that I come here. And so Brad has set this up just right for me in talking about threat models and these issues not being all black and white. And basically, to get at the questions before, there are so many opportunities for us to use quantitative techniques with this data and to try and basically make intelligent disclosure decisions and publication decisions and things like that with genetic data. So we can kind of set the bar where we'd like to de-identify data, so how much data we'd like to remove from a specific data set. And we can also decide and set the bar for where we would like to disseminate it for each different data use, where that should be. So I'm going to talk about privacy implications for rare mutation data. So last year, I think during my segment, I talked a lot about SNP data and familial inference ability of SNP data, but I didn't talk anything about really rare mutation data. And so it's a whole different type of thing. And so there's definitely a growth of mutation data out in the world. There are sequencing technologies getting better and better, meaning that we're basically able to do deep sequencing of genes and also do whole genome sequencing on many individuals. So the PGP, the Personal Genome Project, and other projects are now doing this. And there are also a number of companies that are out there. Not some of these are doing direct-to-consumer genetics, but some of these are actually doing direct-to-consumer genetics with full genome sequences at quite a cost, but considerably more information. And what that means is that this growth of deep sequencing and small resequencing technologies that are being used is that we're going to be finding things that are previously unknown mutations in many new patients. And these unknown mutations, previously unknown ones, are very identifying, and they need to be treated differently than common polymorphisms like SNPs. And so just to give you an idea of what we're talking about here, this is sort of just a map of where each of these different variants falls in frequency in the population. So you might look at SNPs, which by definition are individual loci in the genomes where basically there's a population frequency of greater than 1% for one of the alleles. And a rare variant, by definition, whatever this means, is something where it's less than that. So that's sort of just the bar where that's been put. But then new mutations, for example, might be things that are so rare, unbelievably rare, because they just emerge maybe in the last generation of the duplication of an individual's genome or several generations up in the ancestry. And so we're talking about things that have a population frequency of potentially less than, something in the order of 10 to the minus sixth in population frequency. And so, and repeats are somewhere in that broad swath of somewhere else. So they're not really rare variants. They have polymorphic alleles. Fred Bieber talked about those this morning in some of those examples that he was showing. And they really range in the different population frequencies. And so I'm not going to rehash a lot of this discussion, but there's really broad fear of use in DNA in society. You saw some of those articles that Brad mentioned from last year with the David Craig paper. And so I don't know if you guys have seen the movie Gattaca, but for those who haven't, after sitting through this talk, you definitely have to. But basically the idea is that genomes are deterministic, and you could tell from day one how able somebody will be to do something, every single propensity for disease and ability. And so in the United States, we're kind of trying to get past a lot of these things legislatively. And so GINA, the Genetic Information Non-Discrimination Act, was passed in 2008 and just went into effect this weekend. Basically is a law that's in place now, and state laws protecting similar items exist, that will try and basically not let a company coerce or an insurance company, an employer or an insurance company coerce an individual into taking a genetic test or then making an employment decision or setting an insurance rate based on the result of a genetic test. And other protections that are in place for this data are things like data use agreements, CDAs, and the like, that help protect against attempts at re-identification from investigators or from the public users of a data set. So just making it against the law to try and re-identify somebody. And so those are pretty good, right? Those are things that will help get past the biggest questions, but there are definitely discrimination concerns. Insurance, workplace discrimination, life, long-term, and disability care insurance are uncovered by GINA. And there's definitely all sorts of other ethical issues, as we know, genetic knowledge and personal decision-making, clinical personal decision-making for patients, and also family planning decision-making, implications for family members that might have uncovered information about a family member who's been sequenced and observed to have a rare mutation. And as Fred also mentioned this morning, sorry to sound like a broken record, but the Y chromosome carries so much information. In fact, for maybe 50% of patients can carry your surname. So that's how common the Y chromosome is, that it actually, not common, distinctive it is, that it can basically run in families for such a long time that it carries the surname. And so why risk this whole kind of concern? Why put all this data out there? We definitely want to correlate clinical outcomes with genomic data, and specifically with respect to mutations and rare mutation data, there's a lot of thought in the field, it's certainly not been proven yet, that there are multiple rare mutations that might be contributing to the common diseases that are out there. So why is it the case that these genome-wide association studies that have been done with many patients so far, with statistical power to prove, diseases that we know are heritable, like diabetes and hypertension and heart disease, why is it the case that we haven't found that many alleles from SNP data, really good marker data, in a number of patients that really contribute that same propensity that we know exists from heritability, from family history? And that's certainly an open question, but the idea is that we have to do much more. Sorry, that's an understatement of the century, maybe. But we're going to need to do more deep sequencing, probably. And what that means is that we're going to be uncovering more rare mutations, and that means that more of that will be out there in the ether. And so what we're going to try and do is, in this talk, is quantify what that means. What are the risks that these data pose to individuals and their family members, and how can we preserve privacy, and also even disseminate those messages to patients? How can we sort of tell people, what does it mean that you have a rare mutation? How rare is it? And then how can I kind of internalize that message? And so throughout this, again, I just want to reiterate, as Brett said, there's, this data is so pervasive in research medicine and public health investigations, and it does pose a threat to privacy. It does have the ability to disclose identity in medical conditions and hereditary data, but there's a balance between its utility, for sure, and the risks that it poses. And so I have some collaborators, one at MIT, Pete Solovich, and Ken Mandel at Children's Hospital Informatics Program. And we also got some support from the National Library of Medicine from NIH to do some of this work. The first thing we'll talk about is how to estimate the prevalence of a previously unknown mutation. And so the idea here is that, unlike SNPs and common polymorphisms, where we have some sample, a large number of people that have been sequenced at that locus, to identify what the population frequency of that variant is, here we have a mutation that we've never seen before. And we want to try and estimate what the prevalence of that mutation in the population might be. How likely it is that another individual in one of 6 billion of us might have that mutation. And then the second question I'll talk about is how likely is it that 2 individuals match at that observed mutation. And then the third part, which if we have time, I think we will, is how do these estimates change in different genetic populations? So Caucasians versus people of African descent, how does that change in a different population? And so let's just put this into an example. So make it a little more concrete. You're given a specific single nucleotide variant found in an individual that you can verify is not documented in any other project like dbSNP. It's not a known variant. It's never been observed previously. So you check HDMD, you check all these other sites, you don't see it. So it's different than the reference genome, though. So you know it's something that's a variant. Is it possible to estimate how well one could identify that individual and how would those results differ if that individual is European versus African. So this is the example we'll go through. And just to make this a little more straightforward in the beginning, we'll talk about the case. We'll do this as a de novo germline mutation. So without getting too technical, we'll just say that this is basically a mutation that we've observed just in this last generation. So we'll assume that this patient is the only one that has it at first. And so we'll try and identify how likely it is that a person who has one of those mutations might be likely to find a match in just a small number of patients that's in a GWAS data set, like of 1,000 people. And then the second part that we'll do is if the mutation's not de novo, we'll need to adjust the estimates using population genetics and population size estimates and prevalence. And so some general assumptions we'll start off with, just to keep it simple, are that this is not widely distributed in the population because it is de novo. Another important thing is that we're going to look at mutation rates in the population to try and estimate what we believe the prevalence of a new mutation would be. And so we'll look at different region-specific mutation frequencies in different areas of the genome because they differ throughout the genome. And so we'll use mean frequency values here, but certainly we could use more specific values if we know what the area is. But I'm just going to use this as an example. And we'll also keep it to autosomal mutations and be gender neutral in our analyses to keep it straightforward. We could absolutely change these estimates if we needed to, depending on the patient in question or the individual research subject. So I won't go through this again, but Russ Altman and his group demonstrated that it really, at very low number of SNPs, independent SNPs in the genome, the amount of privacy dramatically decreases. And he also demonstrated how likely it is, and some of the stuff that we'll do as derivative of this work, is how likely two random people in a population will match. And so I guess my point of even putting this graphic on here is that instead of an independent SNPs where the rates of polymorphism in the population are 50-50 or 99-1 for percentages of individuals having an A or a T or a C or G, we're going to have something that's in fact so rare that if you find a match, the amount of privacy you have has basically shifted this curve enormously leftward. And so if you were to publish an individual's de novo mutation, you could potentially be uniquely identifying that patient with just one of those. So just how I'm going to do this roadmap of the problem is, oh good, we're doing a long time. We're going to treat a mutation as a rare allele at a polymorphic location with a frequency P. So these are the P's and Q's from Hardy-Weinberg. And then we're going to calculate that P, the allele frequency of that mutant in a specific population, and calculate the probability of two individuals matching at that location. And so basically the idea here is that we've got this newly identified mutant allele, and we're just going to treat it as if it's any other polymorphism that's existed. It may be the case that many other people have this and we just don't know about it, but we're just trying to make an initial estimate of the population frequency for this newly found mutant variant. And so to do that, we let the population frequency of the mutant variant at the locus be P, as we said, and what I do is I take advantage of the fact that we know some details about the location in the genome where this base pair is. So for the region and the type of base pair it is, what we'll do is we'll actually look at the mutation frequency rates for that area. And then we'll also use a second part, a probability that an observed mutant is a specific type of mutation. So what we'll do is, the idea here is that, so for the first part, the mutation rate for the region of the genome might be transition mutations that occur in an area of the genome called a CPG locus. So that has a certain mutation rate each generation. So each base pair has a likelihood of actually being mutated in each generation with a set number. And what are the things that change that mutation rate? Whether or not it's a functional unit in a gene that might be altered, that might not let an offspring be produced. So there's basically a mean frequency of mutations in each generation that gets altered by population genetics exercises, by evolution, pressures, and other things like that, and then also by corrections. So in sort of biochemistry and molecular genetics, there's all this stuff that goes on that if an A converts to a T, there might be a corrective mechanism that changes it right back to an A. And that happens with certain probabilities. And that's what sort of gets to the second point here. We'll look at the probability of the subtype of a mutation, and that means that we'll look at an example of a conversion from an A to a G. So let's say the reference sequence has an A, and this individual that we've observed with this newly found mutation has a G. What's the probability that an A to a G mutation has occurred in all of the transition mutations that have ever been observed before in that region? And so what we'll do is basically use these two numbers to try and identify how likely it is that this newly observed mutation could have happened. Are there any questions so far? I know there's a little techie. Yeah?
Are they independent? You assume independence, I guess?
Right. So for this part, we're only looking at one locus right now. But yes, in the future, we can...
the region type and the P subtype.
Yes. So we're assuming independence for these two individual things. And the reasoning behind this is actually sound. So the idea is that these are only pieces of this larger puzzle. So we're only going to look at the A's to G's in transition mutations, but the mutation rate that we're looking at is for all transition mutations at a specific locus. So that's the idea there. But using multiple of these, there's a question about independence that's always there. And I think that for newly observed mutations, it's safe to say that you could look at several of them and assume them to be independent, but no one's ever proven that, right? So we can look at the data sources for where we might be able to get these mutation rates and probabilities of having subtype mutations. And so the first thing I did was I looked at a bunch of different papers out there, and somebody's basically used, I can show this a little bit now. So in genetics, it was AMJG, I think, where there's an estimate of mutation rates for different sites throughout the genome that was created, transition mutations at CPG loci, transversion mutations, transition mutations, and all these other types, all other nucleotide substitutions, length mutations, indels. And basically what they did was they used a population genetics model with a divergence time of 5 million years ago, an ancestral population size of 10 to the 4th, and a generation length of 20 years, and common rates of molecular evolution to basically create these estimates of what the mutation rates in different areas of the genome are. And so we'll use these values. And then the other part, for calculating the subtypes of each of these mutations, what I did was I took data from the Cardiff's Human Genome Mutation Database, and we calculated the probability of each of those subtypes just using the probabilities of all the previously observed mutations that have happened that are not at SNP loci, so not at commonly varying loci. So, and those are normalized among all the main type mutations. And so just to show you an example of what that is, we just looked at the HGMD statistics for missense mutations that aren't at common polymorphisms. And so we look at the wild type is either G, T, A, or C, and then we looked at the number of mutations that went from a G to a T, and then we normalized this for each of these probabilities that came in here. So it's this number divided by all of the numbers in this data set. And so we can look at that similarly for all of the nonsense mutations that have previously been observed that aren't at common polymorphic loci, and then all the transition mutations that might have happened and similar transversion mutations. And so the idea is that we've gotten down to something where we have some idea of what the probability of each of these subtypes of mutations is, and we also have some idea of how common each of these locations in the genome, how likely they are to produce a mutant allele in each generation. And so what's the likelihood that you could pick out that individual out of a group of 1,000 genotyped at that same locus? So let's just use 1,000 as a number for a pool, but that obviously can be changed pretty easily with the math here. So technically, the probability that 2 people carry the same allele should include the probability that they both are homozygous minor, so that they have two copies of this rare allele. So let's say it happened a few levels back in the population. That's a possibility. It's really unlikely. And then the other thing is that it could be two individuals matching who are both heterozygous, that they have one copy of the reference genotype and one copy of this new mutation. And so that's the more interesting question anyway, because basically what it gets at is the much higher probability of even being heterozygous. So that's kind of the leading term here. And so we're not going to go into the math, but basically the idea is that the probability of matching for an individual is really led by the probability of really that population frequency squared. And so now we're going to try and figure out what the probability of being the same individual given a match at one of these mutant loci is. And so you can calculate, very similar to the way that Russ Altman's group did and Zhen Lin, you can basically subsequently evaluate the posterior probability of a match at M mutant loci using Bayes' theorem. And so what we do is we just expand this out using a counting version of it. And the probability that a person is subject I, given that they share a set of M mutant mutations, can be calculated directly that way. And so there's some more math in your examples in the book, in the slides that I provided, but I decided to keep it a little more straightforward here. And so for an example, how identifiable is a Caucasian male with a missense A to G mutation in a CPG locus among 1,000 other people? And so with the region-specific mutation information, so we know it's in a CPG locus, we know that it's a missense mutation, and for that, so specifically this one is an A to G, so it's a transition mutation, since it's a purine-purine mutation. So the idea here is that we can just take these values. So we calculated this from the chart of all the missense mutations and transition ones. So we have now a value of 0.386 here times the mutation rate for that area of the genome, 1.6 times 10 to the minus 7th per generation. And we come out to some answer of an estimated population frequency for this new mutant locus. And that ends up being 6.2 times 10 to the minus 8. Not to get lost in the numbers here, the idea here is just that we're estimating the population frequency for a mutation that we've never before observed in the genome. Now with that population frequency that we believe it might have, it's somewhere around that ballpark maybe, right? What we can do is calculate the probability that two individuals will match. They'll be the same person if they both have this copy. So we're taking this pool of 1,000 research subjects, and we're saying, okay, how likely is it that I have this reference person here with this mutant? How likely is it that there might be another person in this 1,000 people that would have it? And the answer is, basically, the way that we phrase the question here, how likely is it the probability that the people are the same person, given that they have one match, is extremely high, because these mutations are so identifying. So it's 0.9993, which effectively you're very, very certain. And so if the probability of one person matching is 1.54 times 10 to the minus 14th, that's the number that we calculated out, the likelihood of identifying a match in 1,000 people is an example of the birthday problem. Have you guys heard of this? Where if you look at the, 65 people, you should be able to, with 50% of the time, be able to have two people with the same birthday. It's kind of like this crazy little paradox. They call it the birthday paradox, but it's really, I don't like using that because it's not a paradox. You can approximate what the probability in 1,000 people of observing at least 50% chance of having a match is. And so basically you would need 14.6 million people to have 50% chance of having a match with one of these mutant loci. And that's, to get up to 100% is much more, many more people. So I think I'll leave the population, we don't have that much time, so I'll kind of jump through this. You can also calculate using a binomial distribution how likely it is that one match or fewer will happen in a set of n people, or two matches or more. And so there, the probability with just the probability frequency that we've observed, the probability of having one or fewer people in 1,000 with that that variant that we looked at, is an exceedingly high number. It's 0.999999998. So the last quick thing that I'm going to talk about, and I think I'll just wrap up soon, is how would these estimates differ in an African population versus a European population, Western European, like a Ceph, or a Nigerian, the Yoruban population, looking at the HapMap samples that we have thought about before. And so one possible approach is that you can use population-specific, region-specific mutation rates to do those things. So look at all of the different variant populations. There are many different databases out there. I know these are really small, but there's an Arab disease mutation database, a Cypriot disease national database, a Finnish disease heritage database, where people are basically compiling for different populations all of the different mutations that have been observed previously, and then looking at what those mutation rates might be in different areas of the genome. So we could use those, or we can go and alter the estimates for different populations, taking into consideration their effective population size and local population-specific heterozygosity rates, and all these different evolutionary and population genetics characteristics. And so I think this is probably a little too technical to get into, but we would expect that a population with more heterozygosity would be less susceptible to having a fixed allele. So you would expect that there's more chance for things to be different. And so that could actually change these characterizations that we've made. And so it should be harder to randomly match two people at a set of polymorphic SNPs or repeats or mutations due to that heterozygosity. And so just in conclusion, I want to basically say, of course, these are not black and white issues. We're trying to quantify things for rare mutations, characteristics of rare mutations, and identifiability issues for rare mutation data that are out there. But it's unlikely, it's extremely unlikely that two people share a rare mutant allele in even any reasonable sized population, and unless they're family members and it's several generations up. And so it may not be possible to preserve privacy in a data set containing rare mutations. And so those things will have to be controlled and changed, either not disseminated in a publicly available data set if we're concerned about the threat model, the Homer threat model that Brad described. Okay, and with that, I'll take any questions. Thank you, thank you.
This is a more general kind of a question. There's been increasing interest in what's called epigenetics, about which I know a heck of a lot, and methylation being an example. Does all of the stuff that you and the other folks have been talking about go through, whether it's one say mutatis, if you include these additional characteristics of the genome?
That's a great question. So the question gets at, how does this change when you consider epigenetics? And I think the answer is unbelievably more complicated, right? So we have this idea that we're not just looking at base pairs, but we're looking at their higher level activities. So something that's happening to change them, either the way that they're expressed in tissues, so the actual genotypes don't change, but some characteristic that happens downstream does change based on some other activity that happens. And so I certainly haven't considered it here, because I think it's, the data are so sparse anyway for rare mutations that it's kind of a challenging thing. But I think that the overall framework shouldn't really be amazingly adjusted. So I could say one thing about this is that this is such a generalizable thing that we've sort of said, oh, we're looking just at the location-specific mutation rate. So if we believe that an area has a higher or lower mutation rate based on epigenetic factors, we could alter that for that region of the genome, or for that specific base pair or something like that. But I think that here it's wouldn't complexify things too much. But I think in the whole overall big picture stuff, it's going to add so much complexity to the GWAS analyses that's happening, and a lot of it hasn't really been considered thoroughly. Any other questions?
So I have a question.
Sure.
Sorry, I got to speak up. I told him I have a question. Policy implications. Basically what it sounds like you're saying is that if something's rare, we shouldn't share it, or we shouldn't make it public. Is that what you're saying? So you.
So don't quote me. I know I'm being recorded.
No. I'm just saying, if you had to go to DBGAP or the NHGRI and you had to give them a recommendation based on your findings, what would it be?
I think my recommendation at this time would be that it should be under a data use agreement if you would want to share this type of rare mutation data. And that data use agreement should specify that an individual should not be attempted to be re-identified in that data set. That investigator should not have a privilege to do that and should be barred from doing that. So my sense is that absolutely this data must be shared, and that's what concerns me about, so you mentioned there in Halpern work where you basically would try and reduce the amount of information, reduce the SNPs that are most identifying out of individuals in a data set so that you could publicly disclose an aggregate data set that contains both genetic and clinical information, potentially like a positive or negative disease. And I'm really worried about that idea. I mean, even though it's probably the only real solution there, right? I mean, it's an information theoretic problem where we're saying we know that these things are so highly identifying, but we want to still reveal these details without actually providing some information. Well, it's not going to work that way. You have to. It's a definite trade-off. So to do that, we're going to basically be blurring out what's most individually identifying about a patient, and potentially losing the clinical and research value from that data set by doing it. So I'm really hesitant to say blurring or removing is the right approach, because I just think that fundamentally data use agreements and laws may be the right solution if it will actually help us get to a real answer on the research side of things. But it's definitely, you know, I still, maybe I'll ask Colin to erase this.
Well, what you're reminding me is I had occasion to be chatting with the Time geneticist a couple weeks ago, and he was all excited about the biology these days. And he's working now with 500,000 SNPs, but with 20 gigabytes of right-hand side data in the GWAS studies he's doing. So he's got the whole genome there. So it's going to include all of these things that you're talking about. So what was your alliterative phrase there, suppressor, whatever? It seems to me unavoidable that these data are going to be identifiable. You're using 20 gigabytes of genetic information per individual with your statistical analysis.
It's not just 20 gigabytes of noise, so there's definitely some unique fingerprint in there. I think that, yeah, it's either suppression has to be used or legislation and restrictions in the way that data are provided. And I think that, you know, I'd always like to err on the side of providing the highest quality information to researchers, if possible, so that they can identify these, in very small populations sometimes, very subtle activities that are going on genetically.
Do you mind if I respond to that as well?
Please, please.
So I take issue with that comment. And the reason why is because, so one thing is that the context is very important for the DNA. So just showing that we have a lot of DNA is not sufficient to constitute a privacy concern. And I think it's going to be dependent on what is accompanied with that DNA. Because just showing that I can find somebody or represent them in a population.
Oh, he's doing it in the context of a hospital healthcare system. He's got all the clinical.
Again, it depends on what he's going to, if he's going to share it or what he's going to do with it. I mean, saying that the DNA is going to be a quasi-identifier or the identifier may be fair. But look, if all I share is if I'm not sharing all of the clinical, if I'm not sharing even a particular piece of clinical information, then it's not a problem. So basically all I'm saying is that the full problem itself needs to be specified. And I agree with you that you can represent somebody, but I don't think that we're going to ever be at a situation where we share an individual's entire genome for research purposes. I think people already recognize that is just not a feasible solution. Sorry, I get combative.
No, it's fair. I think that, but look at PGP, right? I mean,
That's different.
It's a highly consented group of people, right, that understand that they're fully disclosing their data. But I think there are potentially a number of altruistic individuals out there that would want to do that and potentially make them available to researchers. Certainly, as people get older, they are concerned less about these things having a lifelong impact.
How easy is it for a bad guy to get the DNA?
So I think that's it's a substantial difficulty. So, but one of the things I would say is look at where these pools of data are coming from. The publicly available data are coming from Framingham Heart Study, from Jackson Heart Study. These are places, these are towns, right? So you could potentially go somewhere and know where an individual might be and then go get a hair sample or a non-eaten potato chip or half-eaten potato chip, as Fred said this morning. You know, I think that's definitely possible, and I think that certainly criminologists and detectives do this work. I mean, it happens. So, you know, people get these samples without individuals knowing. I think it's a really hard characterization to make that, to say it's easy or hard, though. I think you have to, you know, as Brad said, this Homer attack is like an enormously complex thing to do. You'd have to actually have some handle on the individual's identity, then be able to identify his or her genome, and then you can get some clinical characteristics out of the data set with some probability. And so, I think henny penny, the sky is falling is a great, you know, thing to say at that time, because it's really, it's an extremely hard problem, even if each of the steps themselves could be accomplished with some money. I don't think it should stop us from pursuing research in genetics. Cool.
Okay.
Thank you.
We're done. Chris has to go catch a flight, so we're going to close the session and run in front of the airport.